I will just very briefly introduce this seminar. The seminar has been organized in the frame of the course on biodiversity of mountain resources, and especially for the module on animal biodiversity, because the module on animal biodiversity, of course, deals with wildlife, wild fauna, but in the mountain we also find domestic animals, which are an important part of the landscape and which provide important products and often typical products that can be also strictly related to specific breeds that have evolved and adapted to mountain environment. So today we have invited Professor Paola Crepaldi and all her group of young researchers, all very, very good. I know all of them, and I really appreciate them. So they will tell you about different aspects of genetics, genomics. They are geneticians, so they talk from a genetic point of view about the genetic approach to study these domestic breeds, and they will introduce you to some of the typical breeds of the mountains, talking about ruminants, so mainly about cattle and goats. And in the afternoon, we will go to see a mountain farm and to know some local breeds. So I give the room to Professor Paola Crepaldi from our Department of Agricultural and Environmental Sciences.
Okay, thank you, Silvana. Good morning to everyone. I'm really grateful for this invitation because I think, as Silvana said, that the genetic resources, the domestic genetic resources are really important for the mountain environment. So first of all, I want to introduce my group, me and my colleague, with the presentation of some of the papers that we produce in these years about different breeds and different aspects, so cattle, sheep, and goats, in some researches that are conducted together with national and international researchers. So we do not work alone, but we are a community and we share our knowledge, because especially for animals that have not so much economical value, it is important to have information shared by the worldwide community of researchers. So I obviously didn't, I don't want to read the title, because all these things will be presented in this talk. So the outline of my presentation is this. I will start with the biodiversity concept and the definition of farm animal genetic resources. Then I will focus on Italian genetic resources and in particular ruminant. Then we will shift on mountain with a panorama of the different breeds, and then an indication of the traits and aptitude that are selected and their importance. We focus with an example on about the breeds, product and tradition of the Aosta Valley, and then we will, if we have time, because I tend to speak a lot, if we have time, I will present also the history that is behind the breed that we have now with us in our landscape. So a little bit of history with some focus on the Alps. I decided to focalize on the Alps because we are here in this particular position. So First of all, the definition of biodiversity and farm animal genetic resources. So we know that domestic animal diversity is an important component of global biodiversity, but a long debate was made about the fact that biodiversity have to include or not domestic animal. At the end, also the ecologists recognize that it is important to consider also the very huge amount of variability that we have and that we constructed starting from domestication. FAO indicate that there are about 8,800 livestock breeds of 38 different species in the world, considering also the bird, not only the mammalian, but also the bird. And these species provide a diversity of product and services, as Silvana introduced before. So animal genetic resources are animal species and population within species that are crucial for food production and for the agricultural maintenance of marginal rural areas, and also for the typical landscapes that we have around us. And if you are interested in have a more detail, you can search the database that is known as DADIS, Domestic Animal Diversity Information System, that contain breed-related information on livestock diversity, and that is maintained by FAO. Now, a definition of breed, because we saw before that we have a huge amount of breeds, but what is a breed? A breed is a population within species, and the definition is not only biological. Okay, so it's a subspecific group of domesticated animals that have a common external characteristics that allow to separate them from other similar group within the same species. And this separation could be related to geographical or cultural separation, and have to be accepted as a separate identity. Okay, so is the human that recognize the different breeds. And if you look at the second definition, a domestic animal population may be regarded as a breed if the animals fulfill the following criteria. Is subjected to a common utilization pattern. Okay, so if you select for milk, you at the end have animals that have a particular traits useful for milk production. If you select for wool, you have animal with other traits. If you select for meat, also the conformation of your animal is totally different. Then these animals have to share a common habitat or a common distribution area. A common habitat or a common distribution area means that you have some separation between the different habitats that could be natural or artificial. Natural depend on the barriers, the biological and environmental barriers that are present in a territory. And cultural means that human force this separation. Okay, in which way? Forcing a closed gene pool. So it means that the men do not allow this animal to be mate with other of the same species, and this is one of the condition of artificial selection. Artificial selection means selection made by human that started with domestication. Okay, and the last condition is that is regarded as distinct by the breeders. Okay, so these are the definition of breeds. Then we have different breed classification on the basis of the geographical distribution, for example. So we can have local breeds in one country. We can have transboundary breeds in more than one country. We can have regional transboundary breeds or international transboundary breeds. Transboundary means that is present in different, the breed is present in different country of a single region of the world that is defined on the right in the map that correspond plus or minus to the different continent, not exactly to the different continent, but that is reported by and defined by the FAO. And international transboundary breeds are the breeds that you can find in more than one of these regions. So they are also known as cosmopolitan breeds. Okay. The second criteria could be the adaptedness. So we can have locally adapted breeds. So breeds that have been in the country for a sufficient time to be genetically adapted to one or more of the traditional production system or the particular environment of that particular country. And indigenous breeds or autochthonous breeds or native breeds are a sort of subset of locally adapted breeds. So in a particular country, but in a particular part of that country. And then we have the opposite, the exotic breeds, so breeds that are not locally adapted and that could be that comprise recently introduced breed or continually imported breeds. Okay, so this is the definition. So looking at the number, if we look in that is, the sources that is the database that I showed you before, if we look in that is, we can see that the number of reported mammalian local breeds count at plus or minus 5,000 different local breeds, considering all the species. And considering only cattle, goat, and sheep, we have this number of local breeds that constitute the 58% of the total local mammalian breeds. Okay, and if you look to the total number of the total breeds, this total comprise also the other different mammalian species. But if you look at also to this number, you can see that the vast majority of these breeds are in Europe and the Caucasus, in that region. And this is due to the history of the formation of the breeds. Then we can classify the breeds accordingly with their risk status. And in this case, I don't want to enter in the detail, but we can have extinct, so breeds that are not arrived at this time, or at risk, and inside this category there are some different categories. So you can have critical or endangered breeds, critical maintained if some programs of active conservation is working on this breed or not. And in this case, this is the indication of FAO is related on the number of female and male breeding individuals that are present, or the total number of the population that is present in the territory. But I don't want to enter in this detail, otherwise I have to speak only on this. So what are the number of the ruminant breeds that we have in Italy, plus or minus? Do you have an idea? You can try to guess the number. Please, how many breeds do you think that we have of cattle, goat, and sheep in our territory? Yeah. . Guess is a guess. 60. Okay. For you? Okay, we have about, because the number change depending on what is considered, we have about 130 breeds of goats, sheep, and cattle. Here, there is only a panorama of not all the breeds, and we have local and transboundary, autochthonous, and cosmopolitan breeds that are Italian population of cosmopolitan breeds, because in many cases they are raised in our territory since a long time, even if there is a continuous exchange of reproductors. So it is questionable, this thing. So how many breeds are raised in the mountain? To answer to this question, we have to know the territory of Italy. And so if we look at the territory, we know, and probably you know better than me, that the territory is constituted by mountain for 35% plus or minus, hill 41%, and lowland for 24%. And as you can see here, we have with this color the area of mountain that are diffused, Alps, Apennines, and other Sicily and Sardinia area. Okay, so we have practically in all the reason, maybe not some, but you will have some detail with the work of Arianna, because Arianna is working on this and will present some data about this. So 35% means that almost 35% of our breeds comes from or live on in mountain, okay? Look, cattle. So I count 36 breeds, but also this could be questionable depending on. But if you look at this, if I'm able to show you, there are some arrow here that indicate the place in which these different breeds are raised, but and you can see that a lot of these breeds insist in area with mountain. But if you look more in detail, if I'm able to zoom the presentation, okay, sorry, I'm not able with this. Okay, you can see that we have a lot of breeds that have directly in their name the area from which they derived or in which they are bred, okay? And so you can see here the three strain of Valdostana, so in Aosta Valley. Here, the Pezzata Rossa, red spotted or red-pied d'Europa. Here there is a the Frisona that is not from the Alps, but the arrow I think that will conduce in the lowland, okay? And this is a cosmopolitan, a breed that everyone knows, okay? And then we have also some small breeds like Varzese Ottonese in the Apennine area, and it was known in the past as a mountain breed, Montanina, in another name. But this name is present also in some other breeds of the south, for example, in Sicily, Montanina breeds, typical of the mountain or a particular region of Sicily, and so on. And you can see that also in this part of the Alps, we have a lot of different breeds. Rendena, that derived from Rendena Valley. So each of the different, or not each, but many of the different valley of the Alps have their own breeds. Pinzgauer, Barapustertaler, this is a a breed that is considered cosmopolitan of the Europe, but we have our population. So the Italian red spotted. Then we have here another cow, Burlina, Bruna Italiana, brown, Italian brown, and so on. I cannot insist because otherwise I will not end my presentation. Okay, if we go ahead, I want to show you. Holstein is a cosmopolitan breed, and if you look at the map beside, you can see in red the country in which it's diffused, 9 million head. But if you look at on the right, what you can see, the brown Swiss. The brown Swiss is bred originally in the Alps, so derived from Alps, but is now a cosmopolitan breed because he's raised worldwide and count 6 million animals. Okay, so originated from Switzerland, all the country around the Alps have this cow. If you go in the mountain, everywhere, white, you find these, you can see these animals. And as you can see, you can have also a different shade of pale of color, and large population of this breed are also present in South America, Africa, Asia, and so on. So what it means? It means that the characteristics, the basic characteristics of this breed, I think that there is a slide after, yeah, give to this breed the possibility to adapt it to a lot of different environment, also because it has a very good production. So what are the characteristics that are tied to the mountain environment? Strong feet and legs. These animals have strong feet and legs, have good fitness in terms of fertility, calving easy, the other health, and a solid foundation. So it is really relevant for the pasture in the mountain to have a good locomotion system. And they have a genetical determinate longevity that is also the result from the high proportion of young cattle that graze on the mountain pasture. This is what the Austrian Brown Swiss Association declare for these animals. And as you can see, you can have different breeding objects. So 50% milk, 45% fitness, and 5% meat. Here are the index of the Italian brown, 45% morphology and functionality that is related directly with fitness, and fitness and production, and 55% directly on production milk. But there is also a dual purpose index that recently realized the importance also not only of milk but also of meat in the for this breed. And in fact, a lot of different breeds of the mountain or of the Alps, but not only of different also other parts, are dual, has dual aptitude. So sorry, I don't know what happened. Aiuto. Okay, now the goat. The goat breeds are 40, but maybe after Alessio that worked for the National Association of Sheep and Goat Breeds will present in a better way all these animals. And these animals are typical animals of the mountain. Okay, so not only 1/3, but probably 2/3 of these breeds are typical or are adapted to environmental mountain environmental. I don't enter in detail of the different breeds, or I have to show for the for our yeah no okay. So even for this breed, you can see that you have names that are related with the particular valley in which they are bred. So Valdostana, Vallesana, Sempione, Sanen. Sanen is the name of a breed, but is a name of a place in Switzerland. We will see in a while. Camosciata delle Alpi, Alpina, Verzaschese, Nera di Verzasca, Black of Verzasca, Dilivo, Val Dilivo near Como, Valley of Arianna, the Frisa from Valtellina. Frisa is remember the mask of these animals that on the head have two stripes that are known as frisature in Italian. Okay, and in English I think that is like badger face. That is faccia di tasso as translation. Here, one goat that we will see in the afternoon, Bionda dell'Adamello. So that remember the place in which this Pezzata Mochena, and so on. There are a lot of these. So looking at the name of the breeds will illustrate from which this breed derive. And Sanen, that is the cosmopolitan breeds among the goats, is here you can see the map where is diffused worldwide. And you can see that this goat originates in the Sanin Valley, Switzerland, Alps. So it's an alpine. And so it was adapted to the mountain environment for a long, long time. And then, due to the fact that the breeders, without knowing genetics, I want to underline this, without knowing genetics, are able to have good production with these animals, and then obviously with the with better knowledge also to strengthen this production. And also for this, these animals was exported at the beginning in the USA around the first part of the 19th century, and now is spread everywhere. Obviously, this very high production is possible in intensive system. Okay, so we can have animal genetic resources adapted to different conditions. Sheep are higher in number than goats, are 46, and in this case are not so, the vast majority of this breed is not reared in alpine, but probably in hill, but also in mountain. We have a lot of this breed, as the Rosette in Aosta Valley, Savoiarda in Savoia, I don't know in English, Piedmont region, okay? And so there are a lot, Corteno, Lamon, so there are all for these breeds a name that remember the region or the local place from which they derived. But for the detail of where are bred, you will see in relationship with the mountain, you will see the maps of Arianna that are better than this, more detailed. For sheep, the cosmopolitan one is the Awassi breed that is not an alpine, but is a sheep native of the Southwest Asia. And I want to show you that this breed is widespread in Middle Eastern country, born as a triple aptitude breed. Now is more devoted to milk, but also the other production are present. But what I want to underline is that its spread is related to the resistance of hot climates and the ability to thrive where food is scarce. So what it means that we have different resources, genetic resources that could have particular characteristics that could be useful in different conditions. And so that could be useful also for the challenge of the climate change, and also for this, Arianna will present some detail. Okay, so the biodiversity, so the species and the breeds of farm animals are the product of hundreds of years of human activity in a wide variety of climatic and ecological conditions. And also the biodiversity of mountain livestock resources is a joint result of adaptation to the environmental, that is the longest period of the story of these animals, and then a very short last period of adaptation following the human selection and a more strict human selection. So we now, due to the agricultural industrialization, the mechanization and globalization, have a problem of the abandonment of many local animal breeds as many environments that are difficult to live and that do not remunerate the work of the human. And so such breeds are currently considered endangered by replacement with cosmopolitan breeds that are not so locally adapted as the mountain breeds, and also by the abandonment of animal husbandry, especially in marginal areas. So some times ago, each family have a cow, a goat, some sheep, in order to fulfill their necessity. Now, also in the mountain, the situation is totally changed. So, in fact, there are some estimates of the loss of diversity. And for example, the World Conservation Monitoring Center considered that 25% of farm animal breeds have become extinct, rare, or endangered in the past century. And an estimate from Switzerland researcher say that 80% of the indigenous animal breed in Switzerland, so on a country that is inside the mountain landscape, have become extinct. And these researchers say that probably also in the other region of the Alps, we have this percentage of extinction, because similar agricultural and land management changes over the last 2 centuries affect the entire area. So the existence of local rustic breeds is believed to be necessary to sustain use of marginal areas. But what are the traits selected in mountain? Okay, so what is the history and of this breed? So animal breeds in the mountain were originally selected according to their function. So if you use for full production, that now is not is not a reality, a strong reality, and only those that adapted best to the environment, to the husbandry condition and the demand of their own survived. And in the past, in very hard condition. Okay, so scarce resource for feeding, they have to adapted to steep slopes, narrow trails, snow, ice, relative high ultraviolet radiation, and low food availability. So these are the characteristics that at the beginning this breed have to present. Now, some example of mountain region of Italy. Okay, so this is one of the region with the highest percentage of mountain in its territory, 95%. And here is a panorama of the breeds that are local breeds adapted to that condition. So I think that you know the gray alpine, the Rendena, and Rendena, and the history of Rendena is reported also in some romance of come si chiama? I don't remember. I will say after, but I don't remember the name. Sull'altopiano di Asiago. Rigoni. Okay. Okay, in his romance, is reported that during the Second World period, the breeders were not allowed to mate their animals of Rendena with themselves, but they have to use the brown that give better production because there was a need of food. Okay, and so the breeders that consider this breed better adapted to their condition tried to mate the animals during the night without any telling to the people that they are using their own bulls of the same breed. So the history could change and the objective of selection could change depending on the necessity that change with the changing of the world. So now we are facing a problem of abandonment of mountain areas, a problem of climate change, and so we have to relook at these breeds as a really important source. So Barapustertaler, Pinzgauer, and all the other breeds. The second valley, but the first for mountain territory, is the Aosta Valley. And so here the breeds that are bred in this valley. And I want to show you some more detail on this one, okay? As an example, because if I have to speak of all the single breeds, we will never finish in, okay, 20 minutes again. Maybe I will arrive at the end, okay. So here you can see some very old picture, and if you know the mountain, you can recognize this mountain or not. Mont Blanc. Yes, the highest peak that we have in Europe. And you can see here some cattle that is exactly this one, red pied or black pied and chestnut, that are the three strain of Valdostana goat. And the origin of this breed probably is Northern Europe, introduced by the Burgundians in the 5th century. And this breed have a dual aptitude, they red meat. It is normally, when you describe the breed, you have to describe the coat color and pattern, the shape of the horn, the color of mucose, I don't know in English, mucosa probably, and you have also to report the dimension of the animals, okay? And so male and female. And so here you have 8 tweeters, and these are a quite small aid with respect to the cattle, okay? The breed standard was fixed in 1958, but the genetic study of this breed started with a scientific basis, so with modern knowledge, in 1933 about, okay? What is a breed standard? Each breed have a standard that describes the particular characteristic of that breed, because we saw before the definition of breed. So we need to describe the characteristics, the traits of these breeds. Now, and Matteo, Dr. Matteo Cortellari will show you after. Now we have very strong tools, so genomic tools, to go more in deep, to look if an animal belongs to a breed, if there are really differences between the breeds or not, okay? That could be related to the isolation that these local breeds have. And we can also have a look to the signal of selection of human or of the environment. And in this last period, the worldwide research is devoted to this second topic. So here you have the average production that is reported in terms of milk yield by lactation, the percentage of protein and fat, that are some characteristics that are useful, because the main use of these animals is milk and the cheese, that is the dairy tradition of the mountain environment for whatever, almost whatever breed. The common characteristics are excellent grazing trait, ability to exploit even alpine pasture up to extreme altitudes, okay? And this breed belong to the European Federation of Alpine System Cattle Breeds. So there is an exchange of information and how to manage this population in the best way, okay? Then the typical breeding involves grazing in the summer months and stabling in the winter months. And the most frequent calving distribution is January, November. Then the selection goal is milk and meat quantity improvement. So you have here the percentage of interest in the selection index for the red pied, the red spotted, that are mainly for dairy, okay? And so you can see that you have 50% for muscularity and 85% with a dairy index that comprise a lot of different traits. And here you have that for the black pied and chestnut, you have 10% of these index that are reserved to the combativeness. Why? Why?
We have one question. Can you please use the microphone because so the people that are connected can hear?
Can you go back to the previous slide, please?
Maybe, please. Okay.
Those average production, all the datas, which breed are about?
Sorry?
Those datas, which breed are about?
Is the average breed, the average, yes, is an average, because we have no time to enter in more detail. Obviously, usually these data are reported also for different order of lactation. So you have an average of production for the first calves, the second, the third one, and so on, the parity, okay? But in this case, because we have no time and I want to present a lot of information, I summarized with this data. Okay, and this is data that you can find on the site of the National Breeder Association of the Valdostana cattle.
But
Okay, so if you are interested in more detail, you can find this detail there.
Yeah, no, it was just to understand what was about.
But
Average,
there is
okay.
not a lot of difference among these breeds. Maybe the red pied is producing a little bit more than the other ones, but more or less is the same.
See, also because the introduction of, for example, combativeness as the objective of selection is something that is recently introduced and is requested by the breeders. And so it is needed a long time in order to see the results of a program that you want to, so a direction on the selection that you want in your cattle. Okay.
And combativeness was for the event. Okay.
For this,
Okay.
for the
She's going to show you now.
For the Battle of Queen, that is, I think that you already speak about this.
We have already mentioned the Bataille des Reines because we talked about ecosystem services. So this is a cultural service related to tradition, and
yes,
we had this example.
In fact, if you can look on the right, there is a picture of 1959 of the book that we take with us, if you want then to have a look, about this tradition that is a spontaneous occur spontaneously during the mixing within the same herd or animals when they have to go to the alpine pasture. So a lot of different group of animals were collected together. Okay, and so to establish, but I think that she is better than me on this topic, to establish the hierarchy between the different cow, there was naturally some battles, but obviously human want to have the strongest animals, okay, and so they want to win. They want that their own cow win this battle, and this is now a tourist attraction. And so if you go in Aosta Valley during the summer, there are some of these battles with a lot of public that look obviously are bloodless battle. So no heart, no problem for the cow. But breeders want to breed their animals, in particular the chestnut, so castana, and spotted black, for this. So to have at the end the most combative cow. Okay, and so I don't know, but it's time. And so they want to use this. And in the past, they use, I have not to say this, to tell you this, but it's the story of the breeds. They use another breeds, Arans, that is, have the same chestnut appearance, that is really strong in battles. And in the past, the region and the breeder were have a yes, because they discovered that they are using other breeds, and so they are crossbreeding these animals for these things. Okay, and so this is the story of the animals. I think that you know this really famous cheese, and this cheese is made only with the milk of the Valdostana breed, and has a certification, protected designation of origin, and have a particular tradition of transformation with local enzyme and so on. So there is a so-called discipliner that state in which way these cheese have to be made. Okay. Here there are some data that you can read by yourself. And the Valdostana goat. The Valdostana goat has an herd book in 1999, so recently. But this is not means that this breed started to arrived in this year. Is only the official recognition and manage of this breed by the Association of Valdostana. Have a particular coat color that could have some little diversity, and there are local name for different kind of coat color as in other breeds. And here are reported the characteristics, 8 chest circumference for male and females, weight, and so on. And as you can see, the particular characteristic of this ghost is the particular horns that they have that resemble the ibex horn. And in fact, there were some studies in the past to evaluate if there is a crossing between ibex and this breed, because they insist on the same territory. The selection aims is to conserve the genetic diversity, because for this population that are smallest in number, it is essential to maintain the highest level of diversity and to reduce the level of inbreeding. Because when you have an high level of inbreeding, you have the appearance of pathologies, you have animals that are less strong, and the reduction, and this could lead to the reduction of the survival or the adaptation of this breed. And also for this breed, later with respect to the cattle, I don't know if yes, in 1981 started also the fighting between goats that are divided by weight as in human sport. And the breeder, I remember I studied this goat and I visited a lot of herd to collect the DNA sample for our study. And I remember that the breeder want to have the strongest nail, want to win they want to win the competition and to have the colors that the goat win after this competition. Okay, so it is a question. And as you said before, as you see before, there are some particular products that are related with this breed. So cheese, goat, and also other cheese. These are the traditional dairy product of the Aosta Valley, but also the meat products as the dried meat, mochetta, that could be made with different kind of meat, cattle, or also goat and other. So all these product is represented socio-cultural and also ecological importance for this breed. So in recent decades, the non-material benefit generated by local breeds is recognized as really important. And these benefits are of crucial importance for the development of the territory of mountain region, because they direct influence small-scale land management practice and also the ecological functioning. And this function can contribute to decision-making and developing sustainable land management strategy. And it is believed that especially cultural services that is related to the product, the tradition of transformation are more directly linked to the local breeds. There are a lot of slow food presidi that are particular production, very limited, very localized, made with a particular kind of strain of animals, okay? And so also this can contribute to enlarge the possibility of have a territorial development. I think that I have no time to tell the history. But I want to on a little bit, very little. Okay. I want to remember that if we all are geneticists, in reality, the history of the present pattern that we saw in the breeds is related to a long and complicated history that started with animal domestication. And the animal domestication for the 3 species of our talk today started nearly 80,000 years before Christ in the Middle East from the wild species that for goat and sheep are still present and for cattle are extinct. And so we have that all the breeds that we have now comes from the bezoar or Caparrhea agrus, all the sheep from the Asiatic mouflon, Ovis orientalis, and all the taurine breeds for the Aurochs or Bos primigenius. And what happened? All the animals have the so-called domestication syndrome. So they have some particular characteristics because domestication for the animal was a very strong stress. And these characteristics are floppy ears. We have breed of cattle, of goats, and sheep that have floppy ears. Here, the representation is a dog because it's the first animal domesticated, so the wolf. And a reduced aggression, an increased sociability, and also a reduced brain size. And what happened to these animals? So if you remember the image that I showed before, all the wild species have a particular color that is that has a camouflage importance in their natural environment. Okay, so if I say wolf, you have an idea of what is a wolf. Okay, probably if I say Caparrhea agrus, no, because it is not so famous, but all the Caparrhea agrus have that particular colors that is well adapted to the original environment. Okay, and what happened with the protection of the human? A lot of different morphological, for example, colors appears, and the protection of the men allow them to survive, because in nature they can appear, but they are predated, and so they do not leave progeny. They do not leave progeny, and so the protection of the human after a very strong reduction in number allow to flourishing of different animals with different characteristics, some characteristic in common, and in this way then the human, when a new animals with a particular appearance arrive, say, okay, what a beautiful animals, this is my animals, and continue to mate these animals in order to have animals that could be differentiated by the other, okay? But it is not so simple. This history then of these animals are starting from the site of domestication, are the dispersion of all over the world due to what? To the dispersion of the human, due to what? Cultural, military, political, research market, epidemic, that move the human through the world. What happened in the Alps? In the Alps, the same. The continual human settlement in the Alpine area began toward the end of the last ice age and was consolidated during the Bronze Age, as testified by the Rupes painting that you have here. I never saw in person. I want to show it. Maybe you you have. And the famous Mummy of Otzi, that is so-called also Iceman, that lived approximately in this in the same era. By that time, most of the population has already shifted from hunting to and gathering economy to the practice of agriculture and livestock breeding. So the action of the human started from the domestication. And obviously, these men really do not know genetics. And so they select the animals because they choose on their own decision, on their own needs, what animal to bred and what animal no. And in fact, the number of the animal domesticated are really small with respect to the number of wild animals. Okay. Then, as for the others, romance, war, conqueror, and so on, shape the animal diversity. Why? This is a monument that is present in France, Tropheum Alpium, that commemorate the victory of the Romans against 46 tribes in the Alpine Champagne. And if you look at the name of the population, I want to underline the second one, Camuni. And so if you read the name, are names that are reported also in the valley that we have, and in the name of the population. And in fact, in the past, one very famous genetician studied the name, the gene, and the population in order to study the diversification of the history. And so all these different tribes have probably their own animals selected and isolated in different valleys. Okay, so maybe some exchange was present also at that time, but geographical isolation leads to the productive isolation, and so to accumulate by the so-called genetic drift, by random variation, different traits in different population. And then what do the Romans? What is one famous things that the Romans do? Constructed roads. Okay, constructing roads means put in communication. And so they constructed roads toward the Alps. And so the isolation was reduced. And so the history of the evolution of the world is a continuous reduction of isolation, even if in this time we are coming back. Okay, and so the globalization is that you can go whatever you want in a very quick way, maybe not so sustainable, but you can. And in the past, the history was not this. And this history is until recent. So it was a very recent part of the history of the population. Then, so you can look at this in this way. From the prehistory until the 18th century was a very low change that accumulates in animals that live with humans. Then what happened? Then there was the fashion way of breed the animals that is made by some pioneers in England. The person that are represented in slide, Robert Beckwell and Robert and Charles Collins. And what they do, they follow the same rules that the Arabs use with the Arabian horse, because they imported in, not in this period, but before the horses, and copy the tradition of these tribes. And what is this tradition? Is to careful choice the breeding stock. Is register by an oral tradition at the beginning the pedigree of the animals. So they tell to inside the tribe, they know exactly the pedigree of their animals and the maintenance of poor lands. They do not want to mate their own animal with the animals of other populations. So following this, they started to select animals that are equal. And in fact, the first breed that have an handbook is the shorthorn that they selected. This is a typical animals for meat production. And they was lucky because selection for meat traits is more easy because have a strong hereditary basis. Then in the 19th century, the discovery of Mendelian law give more emphasis on the, thank you. I finished. I leave this, okay? Here are the data of the different handbook. No. Then statistics comes and allow to explain the frequencies of gene and allele with mathematical statistic theory and pose the basis of the modern quantitative genetics that is applied to the population. So we understand better the way to inherit the traits and the variation of these traits in the population. And at the end is my last, not exactly the last, but and your first. Now we are in the genomic era. So now we have new tools that allow us to better investigate the variation inside the breeds and inside, between, and the different breeds. And we can relook at the history and found genes that are associated with human selection or with the adaptation to a particular environment. And so you have a small bibliography, and Thank you for your attention. I'm sorry for how long the presentation. Facciamo quiz? There is time for a quiz. We want to evaluate your knowledge about the first cattle breed. So it's only to know if you are able to recognize some breeds, but this is the easiest. Then we have goats, and then we have sheep. But for the sheep, it's quite difficult actually, because the variability is less pronounced. Even if the expert could recognize all the different breeds, I think no.
Okay.
I leave.
Yes, I'll try because it's the first time I use them. So, okay, you have a spoiler. That's okay. Present. Okay. Okay. So you should be able to link to this. I don't know what. Okay. You can go to menti.com and use the code that you see on the top of the screen. And then you can put a nickname, and you can do it also if you are attending online, if you want. You can choose a nickname, and when you're ready, we can start. The code is 17767926. Devo nascondere i nickname, quindi perché sono i buchi.
Okay.
Okay.
Yeah.
You're all in? Yeah? No? Okay, perfect.
So someone. Okay.
Okay. Il best sono i nickname di solito.
Okay.
Okay. Okay, I will start. Okay. Oh, okay. No worries. I don't know if the ones online are connected, so I don't know the number. It's just a 5-minute quiz, so don't worry. Tiger is back.
Sono io, ho messo in prova.
Ok, tu sei da te. Vuoi farlo anche tu? Perché non sai quelle come.
No, io l'ho fatto.
Vuoi fare il quiz?
Poi sarò breve.
Vabbè. Okay. Are you ready? I'll start. Okay. The first, the highest. Okay, so you can see the image, and then you will have 3 possible answers. Choose the correct one. Yeah. Okay.
Try to guess the right one.
Yeah, because otherwise we spend a lot of time.
Okay. And what is the answer?
The answer was Chianida. So from Tuscany, one of the main beef breeds.
Not of that.
Not of the Alps. So Daniele is winning.
Okay, ready? Okay. This is easy. More or less, because I put the same ones. Great. Very nice. So we will see the new. Oh, tiger baby is winning now. No, Daniele is safe.
Yeah.
Yeah. Because you have to make sure. Okay. Yeah, we are the best one, among the best one. Look at the muscles. Yeah, Piedmontese. They have the double, yeah, in the
Doppia
coscia.
coscia.
Yeah, la doppia coscia. And so they had some problems with fertility and calving, and so they are strongly selecting for having small calves at the birth so they can deliver easily.
They have a good.
Yes, now they are succeeding in the, yeah,
Strong problem.
yeah. They have done a great work. Okay. Okay. And Daniele is always at the top. Okay. Okay. This is easy. Okay. No, two have the wrong choice, but already I chose them. Okay, we have seen the pezzata rossa, so Sil was the fastest. Tiger baby and Daniele are near. You're tiger baby.
Not tiger baby.
Okay, we are 5 out of 9. Okay. You know it?
No.
It's easy.
Ah,
Because
okay.
it's the only
Okay,
one.
yeah,
with-
the only one.
With this pattern, it has a particular pattern. Okay, almost. So everybody, good. Very good. This was from Trentino Alto Adige. Daniele, always very fast. Okay. Oops. The system doesn't know that you did wrong. So. Good. Tiger baby, you have to be fastest. Okay. Okay, this is really easy. Okay. 2 of them were easy. Yeah. So. No, 3 people made it wrong. This is the Olsen, that is the cosmopolitan breed. Yeah, that most widespread. Okay. Okay, almost. The Rossa Mediterranea is not pied and is from Sicily. And, well, she's not pied. Tiger baby It's not the first one. Daniele has maintained his position. Oh, last one. Yeah, indeed. I remember the one that. This is one of my favorite ones. They are stunning in person. It was quite difficult, and it was a maremmana. They have these huge horns. They are huge dimensions.
At the end of the 3 years of today, we will give you some suggestions where to some read about to a better knowledge of the.
Yeah. Daniele is the winner. So my compliments. I don't know who is among the people that.
La casa, c'è una discrepanza.
Very good.
Thank you very much.
Oh, thank you. Okay, so we
Can I
will
say
have.
2 words? Can I say 2 words?
Sorry?
So can I say 2 words to Professor Crepaldi, because I graduated in 2010. I'm from online, of course, and I'm graduated 2010 at the University of Milan, and and right now I'm a professor at the high school in Piedmont, in Crodo, that is close to in the north of Piedmont, and I had the like teacher, Crepaldi professor. So this is the reason that I won.
Thank you, Daniele.
Thank you very much for all your knowledge that you transmit to us. And this is I thank you also, because there will be a good occasion for my student do a lesson with these techniques that I didn't know. So thank you very much for the suggestion.
Okay, thank you. Okay, now.
Okay, see.
Now, Dr. Matteo Cortellari will introduce some tools, genomic tools that could be useful for breed conservation. We will wait some minutes to start.
With Dr. Matteo Cortellari, who will talk about genomic applications.
Hello, everybody. Now, as introduced before by Professor Crepaldi, we are now in the genomic era, starting from the year 2000, when the Human Genome Project was completed, and the first genome of the first species, that was the man, the humankind, was totally sequenced. We start to sequence all the other species, and as the first species in the domesticated animals, we start with cattle. That was the most important, with the highest value, and give the start to all the study that we are doing today on domesticated animals. And because we started to know the complete sequence of the genome of single animals and population, and we know the mutation that are in the genome, and we can study which mutation, where it is, and which genome is in, which gene is involved, and what is the the purpose of this gene, and what happened when the mutation happened, of course. And after that, the second species we sequenced was the sheep, not in 2017, because here in 2017 we have a second project of resequencing, because we start with the first time that we sequence a single animal with the complete genome, as in the cattle in 2009, but each year or each now is very very close schedule. We have a new resequenced genome of different animal of that species, because we can have all the spectra of the mutation that the population can have. And in this case, we have a new version of the sheep genome 2017 that was completed, because we have the necessity to compare with the wild species that maybe have the same number of chromosomes, but have different mutation, and to understand the the evolution of the breed and of different phenotypical characteristics, like in this case, the number of the presence of the horns, or the numbers of the horns that the animal has. We need to continue to have new new resequencing to know all the possible mutation in a species. The last, we can say that the last species of ruminant domesticated species was the goat. In this case, maybe was the one with the less value, and in this case, we need a big consortia of university and institution to share the effort and the money to have a complete sequencing. And now, from this complete sequencing, we have, we can say, the most interesting tool that are this NIP chip. Okay, this set of specific mutation of all along the genome of the species that allow us to have a pretty complete view on all the genome, and understand which are the mutation or the modification that are in place in that species or in that breed, and are the tools from which we can start to study all the modern all the modern breeds. And indeed, this NIP chip is a set of thousands and thousands of mutation of single nucleotide that we have in specific region and in specific place of the genome, that change maybe from one breed to another. In this case, they are a specific chip for each species. And then maybe in goat, we have different breeds, and in that SNP, they have different mutation. Maybe Nera di Verzasca has a C, but another breed, like Bionda dell'Adamello, has a T. And then we go to compare this mutation. And starting from that, we could do a vast number of analyses. And the first and most important, as we can see here, we can define the genetic and the genomic background of one particular breed. We can identify what is the unique characteristic of that breed in terms of his genome, and compare to all other breeds of this species, and understand if they are really different or they are close, maybe closely related. In this case, in this graph, we have different color. Each color represents, we can say, a breed. We can say the name of the breed down in the graph, and maybe we have example like the Eurobica is in the yellow color. We can see that all the animals have the prevalence of this color, but some animals have other components, maybe green, and this green is related not only to Eurobica, but for example to other like Valpassiria. Or
may I
yes.
add something? A single animal is a vertical line. Okay, so the group are different animals of different breeds and are represented all together. And so you can see, as Matteo said, in yellow, the Orobica, but you can see also that some animals in Orobica have a component that is green, that is typical of other breeds. And so in this way, we can distinguish, and only the,
Yes.
I want to precise
Oh.
the fact in which they can read the single animal.
The single column.
Sorry.
Yes. And yes. Genetic is all the study of the genes and the phenotypic expression. Genomic is the study of the genome, of the sequence of the single nucleotide that we have in a species.
Yes. But you can use also in a interchangeable
Yes.
way. Okay, so
Now,
now when we
genomics
speak
is
of genetic,
compared-
genetic or genomic background, okay, so it depends on the tools that you are using. But when we speak about background, we are speaking about a genomic. So the evaluation of the genetic composition of a breed, but through the use of genomic tools.
Evaluating all the sequence.
All the sequence as Matteo said. Okay, so more in depth.
Yes.
Question about the genomic shared. Of course, we select some genes that are shared between lot of species and to have a measure and of their of the genes that are not shared and typical of or some lab races are selected some unit of measure and we see they are shared.
I think that you will understand better this with the next slide and the presentation of Matteo. Because the SNP chip that we use, so the huge amount of mutation are, as Matteo said, in a particular location on the different chromosome of an animals. Okay, and so we have the genotype at all these points for all these animals that we can evaluate. So all the goats, okay? And then we compare them, but are not genes, are only mutation in the genome, okay? And looking at similarity or dissimilarity, we can arrive at the end at this graphical representation. And so we can have an idea if we want to know, but I think that Matteo will show you in the next slide how to see the human selection, different human selection and different environments.
Yes, basically the comparison between different species might take in account also the differences in the number of genes. In a single species, the DNA is, with the exception of some structural differences in different individuals, it's more or less the same. So you go to see the basis in that position for all the individuals. And you expect that the vast majority of individuals has a specific gene position, but all the individuals are genes in that position. So you're not interested in this kind of studies in the genes, but you are looking at the difference in the whole genome. Whereas if you want to understand the difference, and he will show it within specific traits, for example, you then can go to see what is the gene that include that mutation. Okay.
Yes.
And also, if you want to discover the genes, you can compare, for example, animals that are bred for milk or meat, compare, and then you can have an idea of the region of the genome that differentiate these two kind of production. And so you can evaluate what are the genes inside that region that are probably responsible for the different aptitude of the animals, or different color, or different horn, as Matteo showed before.
Yes. In this case, we have, for example, this tool could be used for all the species. In this case, we have applied on cattle, and we can see not only maybe the group of Italian breeds, but all over the world, we can have a proportion of the genome that is shared between all the population and all the breeds. And maybe coming from the breeds coming from Africa, I have a particular color that's shared between them and not between other populations that are from Europe. And in this case, we can have an idea of the differentiation of this population and the uniqueness of the population from the geographic region from which they come from. As we can say before, we can have each line of the graph that we have seen before, as Professor says, is a unique animal. Okay, and we can assign that animal to a specific breed because of the color of that animal, because that analysis lays on an algorithm that analyzes all the SNPs and says that the animal that had this kind of mutation are in this breed, that the other animal are in this breed, and assign a color to each breed. And then we have maybe animals that are, we can say, perfect representation of the breed, that have all the medium mutation that the breed have. And maybe there are other subjects that have half yellow, half green, or 2/3 yellow, 1/3 green, because that green is belong to another population. And maybe they share this part of mutation, maybe not because their parents are exactly one from a breed, one from the other breed, but maybe the granddad came from the specific breed, and they carry that mutation with them. And then we can have the image of the population and of each email. And then if we compare all the population, the single subject in this case could be assigned based on his color to his population. SNP are the mutation that we are studying. Okay, in the SNP chip, we have the single mutation from one nucleotide to another. Okay.
And the name stay for single nucleotide polymorphism. So a difference of a single base.
That is C or B or T.
At the same time, we can also see that with other analysis resemble the geographic location and the geographic distribution of our breed. In this case, we analyze all the SNPs that we have in our chip, and we can see that in the image on the left, my left, that is your left, yes, have all the single location of the breed. In this case, they are sheep breed, and each point is the position where the breed is raised. And in the other, we have all the sample of the animal of this breed that we have analyzed. And we can see that the position, the distribution in the graph, okay, resemble the distribution along the peninsula. For example, we have the Valle del Belice, that is the green one in Sicily, that stay in the lowest part of the graph. And for example, we have the Biellese that is in the north and stay in the part of the Alps. And they can be differentiated because, as we can see I think in this slide, all the, that is the same analysis without the map of Italy. We can say that we have the group of the breed of the north that go all together. And then we have a big group of the south, of the southern breed, in this case, our goat. But we can distinguish from this analysis the breeds of the north because they share a specific pattern of mutation of SNPs, and they can be close together. And then the breeds from the south that have a little bit different. Then if we compare, maybe these are only Italian breeds. If we compare with all the breeds of the world, maybe they will be very close. And we say Italy, yes, they will be a single point. All these breeds will be in a single circle, because there are breeds from Africa that are completely different, and this change the image that we see. But if we can scale to the dimension of Italy, that in this case we can see the difference that are not that follow the geographical pattern. Now the point is, because we can, this is a we have the map and the analysis that we have seen before. That
the distribution of the points is according to
the analysis,
similarity, the genomic similarity. So the closest points are similar from a SNP and genomic point of view, whereas the farthest one are different in terms of genomics. So you can see that breeds tend to group together, so that the points with the same color are very close together. And if we rotate the graph with the points, we can see that basically it follows the geographic distribution of the breeds.
So,
Yeah.
So,
based on
yes. In synthesis, the genetic distances are very, very close to geographical distances. And so if you are speaking of the distance between Edolo and Milano, with respect to, I don't know, some Edolo and Bergamo, okay, you can appreciate the difference. But if you are looking at the distance between Edolo and Milano and Edolo and Bergamo, and Edolo and, I don't know, New York, so it is all relative. And so the distances between Edolo and Milano and Bergamo are more.
Geographical parallel to the genetic is quite spread phenomenon.
Yes. And if you do not find this, you have to examine what happened in that population, in that animals, that probably are not always bred in that particular area, but maybe arrived from other area.
For example, we are studying the goats worldwide, and we can see that they basically are, okay, thank you. They are basically separated according to the continents. But for example, we have some breeds that are on a specific island that were imported from Africa. And so they are very similar to the African goats. Or for example, we have a breed that is called Norwegian, that is bred in Africa, but is between African and European goats, because it was imported obviously from Northern Europe. So you can also understand the history maybe of the selection or of the importing of animals, of breeds, according to the genomics sometimes. And you have to go back and see if effectively there was an exchange in animals at different places that you can see also genomically.
In this case, the analysis is starting from the same tool, but it's different because it's phylogenetic reconstruction, that we can say which breed come from which breed, okay? the phylogenetic tree we have here in this case. And we can see that we have a strong branch that are the breeds of the north that are in green, that groups all together. And then we have also the contrast with the wild and feral animal that are the roots, in this case, of our tree, that are the Monte Cristo wild sheep, goat that we have. And maybe there are other breeds that are maybe coming from the same region, but are not so close related as in the north, because in the north they share a specific pattern of SNPs because of the traditional raising technique and breeding technique that we have in the valley, and they not closely related, but they can share some of their mutation. These are the same, we can see, disposition as we have seen before, but in this analysis we can see the proportion of this, the genome that, the mutation that the breeds share one with the other, and each part of the circle is a breed. I don't know if you can say the name, maybe, and then we have all the line that that share and bond one breed with the other, that can be more or less thick, maybe, and this is based on the proportion of this of the haplotype of the series of SNPs that these two breeds share one with the other. And we can see that the green one are the Alps, and some breeds share their haplotype and their SNPs only within other breeds of the Alps. Maybe there are others like the Valdostana that we have seen before, that is very unique. It doesn't share this haplotype with other breeds. And maybe in the opposite, we have the breeds of the south or in the central Italy that are in red and in purple in this case, that share a vast majority, a big proportion of their haplotype within all the other breeds in the same, you can say, region area, because of the difference in this case of the traditional breeding system that we have in the Alps, a vertical transumanza, abbiamo l'alpeggio, and the traditional transumanza in the south goes valley from valley, and then the animals, maybe animals of multiple breeds, share the same pasture, and then in this case we have a larger exchange of mutation, because animals of different breeds may be bred together and crossbred during the years of the past time, and we have a larger sharing of the haplotype. One analysis very important in this last year is represented by the runs of homozygosity. These runs of homozygosity are a series of these SNP markers, okay, a region of the genome that are inherited from the parent in homologous situation, because we can say that they are the same, okay? We have a strand of this SNP that is identical, okay? In one animal of a breed with all the other animal of this breed, or only a small group of this animal, okay? They are they have this homozygosis status. I don't know if you have an idea of the homozygosis. Okay, they are the same allele at the same locus, they are identical, okay, both coming from mother and from the parents, okay, they have the same identical allele, and we have a series of these markers that are identical because they come from mother, from the parents in the identical state, okay, and we have this region in the genome of the animals that are exactly the same. You can see we have the same series of mutation and nucleotides. These are particularly important because based from these runs of homozygosity, we can evaluate the level of inbreeding of an animal. We can evaluate the pathology coming from their parents, their hereditary genetic disorder that we have, and also we can have an estimation of the difference between the population based on these different regions of homozygosity that the population share. We can see in this case, in this graph, we have one chromosome of one population, and each line is an animal, okay? Each row is an animal. The line that we see are the proportion of that chromosome that in this animal are identical because they come identical from his parents. And maybe we can see that there are regions that are shared among more than one animal. And we can see that that region is typical of that breed maybe, and that we have to understand which gene that region contains. Starting from this row, as I said before, we can evaluate the difference. We can evaluate the inbreeding that a single animal have. Okay, and we can compare the inbreeding coming from this row, coming from genomic analysis, with the inbreeding that we have from the pedigree of that animal. And in this way, we can, if it's necessary, we can correct the values of the pedigree because it's difficult also in species like goat and sheep to assess the parentage, maybe because the animal jumps from one not pasture
Jump the fence.
yeah, yes, jump the fences and the different group and mix together. And we don't know exactly the father is that animal or that, and maybe using genomic, in this case, we can assess exactly the parentage and the values of inbreeding because we know all the parents. And this, we can correct the mismatch that we have in the pedigree and have a better estimation of the total value of inbreeding of that breed, because it is one of the most important threshold that we consider to have an endangered breed or not. This is the image that we have seen, and the mismatch, because in red we have the animals that are not not correctly mismatching the association between the values of the pedigree and the genomic. The other very important, we can see here, work done only on the goats breeds and population of the Alps, in which we have evaluate this value of inbreeding. And we can see each point in this case is one animal, okay, each color one breed, and we can have very strong difference in the population. For example, we have the Valle di Livo, okay, that have the lowest values of inbreeding. That is good to have a low level of inbreeding, but in this case, we know that the history of the breed, and we know that in this breed that is in a valley near the Como, the Lake Como, all the animals were collected and maintained in population. Maybe also the animals coming from other breeds, because it's a very poor valley and the system not permit to exclude many animals. Okay, they tend to retain all the animals in their flock, and this caused a reduction of the inbreeding because we mix more breeds together, maybe. Okay, and in other breeds, like for example, the Valdostana, we have these 3 points that are on the top of the graph that are the highest value because maybe these 3 animals are closely related that was born from breeding from parent or animal that were directly related one from each other, and and this is is not the medium value of the breed, okay, because we can see that the medium level of the breed is not so high as the 3 animal, but we can underline which animal have value, very high value, or they influence the mean value of our population. This same value can be analyzed starting from the length of the run of homozygosity that we have seen before, because we know that the longest runs of homozygosity are inherited from their parents, okay, from the breed with parents that maybe are closely related, okay, and they share the same strings of allele, and then in the progeny we have long region in homozygosis, okay, maybe and in the opposite we have the shortest, okay, runs of homozygosity that we have seen before that are originated in the past, okay, maybe 2 or 3 or 20 generations ago this raw originated, and the length of the raw changed during the generation because mutation happened and they break the length of the raw, and they start when originated is long and maybe all the generation becoming shorter. And in this case we can see the proportion of long raw and short raw in each breed. In yellow we have the longest, that are the more recent, and in green we have the shortest. In blue we have the shortest, while the middle are red and green. And then we can see maybe in breeds like, for example, the Valdostana or the RGGs, the Roccaverano breed, that have a big proportion of yellow and long raw, maybe in the actual generation, the actual population, we have a lot of breed between animals that are closely related, and then we are increasing the inbreeding, okay. Maybe in other population, like for example la Bionda dell'Adamello, we have very balanced proportion of this length of the raw. That is the last column that we see. And in this case, maybe we have not so low value of inbreeding, but is correctly managed because we have short proportion of raw that are long and then are inherited in the population by the breed of close related parents, okay, and this is very good practice in the management of the population. Yes. The FROH is the terms to indicate the genomic inbreeding. Okay, we have, in this case, we have we have F PED, that is inbreeding from pedigree, okay, and F raw is inbreeding from runs of homozygosity. Okay.
F means inbreeding, so F raw or F PED.
Inbreeding is the species of Valdostana. And if you breed them, you got them.
Breeding? Inbreeding means that you are mating animals that are parents. Okay. They are related. Okay. They are strictly related. So, yes, it's not a good thing, and depend also by the dimension of the population. Okay, so in small population, you have only that animals. There are some strategies in order to reduce this level of inbreeding. So we do not want an high level of inbreeding, and so we have to study the mating system in order to reduce the inbreeding that is present in this population. But if you do not know the pedigree, you cannot know who is related with who. Okay, and so with these tools, you can have an idea, a more precise idea, even if you do not know what are the parents. Because for cattle population, you can come back until 10, 12, 20, 25 generations. So I try to write the name of your ancestor, your grandfather, 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 grandmother. You do not know, you do not have all this information. But also for some breeds, you do not have this information that are very important in the manage of the conservation of the animals and for the breeding of also animals that are under selection.
They are marked.
They have an ID, a marker.
No. The farmer has to divide them and to select the fathers and mothers to be bred together.
So mating is decided by the farmer. I mean,
Yes.
In the wild,
in the wild, they have a lot of communication tools to identify each other because they use sight, they use smell, they use vocalizations. So they have different cues that allow them to identify each other individual.
Normally with natural matings, farmers do mating groups. So each groups have one male and 20 female. And in this case, the male is one, and so you know the father. And when the calf or the kid born, you know the mother because the mother allattare. Yeah. Yeah.
Sure.
The problem is when the farmers don't do mating group. So you have more than one male with more female, and in this case you don't know which is the sire and the dam. And this is a problem because you can't estimate inbreeding, you can't estimate breeding values, you can't manage a population. And genomic tools help us to.
And in order to be enrolled in the health book, you have to know the parents of the animals. And so all the important animals, not all in reality until recent period, are evaluated with DNA in order to test the paternal, who is the father, who is not the father. And after these indications are recorded. And so if you want for each single cow, you can have all the pedigree and all the information that are certified with different tools. And now you can use molecular tools, maybe not SNP, but other tests. And before other tests, before the era of genomics.
in this case, different we are going to evaluate the difference in the footprints that selection leave in the different genomes. To understand, not exactly, there are methods to understand which is artificial selection made by man that is more recent, and natural selection that maybe go beyond thousand and years and have a different impact on the genome. In this case, in this graph, we have used runs of homozygosity to identify regions. Okay, each part of the circle is one chromosome. Okay, each line inside the circle is the result of an analysis, and we have underlined the highest value that are in red and not in blue. Okay, and that are shared multiple analysis. For in this case, we have divided the breed from the south and the breed from the north to understand which are the breed that maybe the genes that in the region of the genome that are typical and have maybe information from the breeds coming from the northern Italy differently from the south, which are the difference, which are the genome that maybe can have more information, okay, for this group of breeds. In this case, we have seized that I will talk only about the breeds of the north because there is no much time.
All these examples are probably a bit difficult to follow because I don't have a strong genetic background. So maybe we can go a bit quicker on these to leave a space for the other two speakers, because it's already 11:15. So maybe we can try to speed up a little bit.
But in this case, we are genes that are related to fertility and pigmentation that are typical from breeds of the north. We have to find out. Another study in this case was related on the aggressive, the patterns of aggressiveness in goat. We have studied on the Valdostana goat and the Bataille de Chèvre, and we have seen that the goats that are selected for aggressiveness are the dominant one, and in this case they are related to a strongest immune system. Okay, in genes we have found genes that are related to the immune system that are more present in the group of the animals that are dominant and you can say that win the battles. Okay. Lastly, the first and most important tool that we have are the genomic values. The genetic and genomic values of the animals that are used to select the animal to breed in the different breeds. Okay, maybe they are more used in cattle, okay, because they are animals of high value, but today they are spreading to all other breeds. Also, we can see with Alessio in sheep and goat, and we can use this value that we can calculate on the animal before he was born, practically. At the time of his birth, we can have the value that he can give to our population, the increasing that he can give, because we can analyze the DNA and say, yes, this animal could improve the production of our population or not. And the first.
I want to add a detail that with genomic tools, we can evaluate the breeding value of the animals once they are born, because we have to analyze their DNA. But previously, with statistical model, we can estimate it basing on their progeny, their ascendant, and so on. And so without, in reality, looking directly to the DNA of that animals. Okay. Thank you.
Thank you very much. Adesso facciamo 5. Ma sono le 11:15, Cioè, devo parlare in 2. Vedete voi.
Io provo a far veloce.
Okay, so maybe just 5 minutes because this presentation was not easy to follow, I understand. So maybe 5 minutes to reset our brains and then we start again, okay? Is Alessio Negro, who is a PhD student at the Department of Agricultural and Environmental Sciences under the supervision of Professor Crepaldi. And he also works for ASSONAPA, which is the National Association of Shepherds. I don't know how you... Okay, sheep and goat breeds. Okay.
Hello, everyone. Thank you, Silvana. As Silvana said, I work for the Italian Sheep and Goat Breeders Association, called ASSONAPA, from the Italian name Associazione Nazionale della Pastorizia. ASSONAPA manages more than 100 sheep and goat breeds recognized by the Agricultural Ministry. ASSONAPA has the aim to protect the livestock at the national level, and it do this through the animal breeding and biodiversity conservation of the Italian small ruminant population. Furthermore, through the promotion of research, dissemination, and new application of new techniques. ASSONAPA manages around 350,000 animals, reared in more than 3,000 farms. And ASSONAPA has the general office in Rome. And furthermore, we have a genetics center where we rear two dairy sheep, Comisana and Massese breeds. Okay. Here you can see the list of the Italian sheep. Italian breeds are divided into breed subjected to selection program and breed subjected to conservation program. And in the table, in the first table on the left, you can see the breeds subjected to selection program, and in the other two, you can see the breed subjected to conservation program. In yellow, you can see the breeds reared in Lombardy, such as Bergamasca, Brianzola, Pecora Ciuta, Pecora di Corteno, and Schwarzen-Nasenschaft. The number of animals of these breeds ranges from around 3,000 animals for Schwarzen-Nasenschaft to around 3,000 animals for Bergamasca sheep. All these sheep are meat sheep. Here you can see the list of goats, of Italian goats. In the first table, you can see the breeds subjected to selection program, and the main two breeds also reared in Lombardy are Camosciata delle Alpi and Saanen. And in the other two tables, you can see the breeds subjected to conservation program, and in yellow, you can see the breeds reared in Lombardy, such as Bionda dell'Adamello, Frisa Valtellinese, l'Arianna orobica, and Verzaschese. Before to see which is the role of ASSONAPA, we have to talk about the law, the Italian law, and in particular of Decreto Legislativo number 52, that is the discipline of animal reproduction. In this law, we have three main novelties. First of all, a new definition of the breed concept. On the right, I reported the definition. A breed is a population of animals sufficiently uniform to be considered to be distinct from other animals of the same species by one or more groups of breeders which have agreed to enter those animals in breeding books with detail of their known ascendants for the purposes of reproducing their inherited characteristic by way of reproduction, exchange, and selection within a framework of breeding program. I think in this definition there is two key points. The first is that an animal to be belongs a breed, it must be enrolled in an herd books with known ancestors. And the second key point is that its reproduction must happen within a breeding program. That it could be selection program or conservation program. The other two novelties is a new organization of the Italian breeding system and a new definition of herd book. Here you can see the organization of the Italian breeding system. On the left, in the scheme, you can see the old organization with the old law number 30 of 1991. And on the right, you can see the currently organization of the breeding system with a new law, Decreto Legislativo number 52. In the currently organization, the breeders is member of both Associazione Nazionale Allevatori and Associazione Nazionale Allevatori. The Associazione Regionale Allevatori are member also of Associazione Italiana Allevatori, and together they have the aim to collect data into the farm. Instead, Associazione Nazionale Allevatori is called in Italian Ente Selezionatori, and they have the aim to manage the Italian population. Furthermore, Associazione Nazionale Allevatori are member of FEDANA, Federazione delle Associazioni Nazionali Allevatori, that groups many Associazione Nazionale Allevatori. Here you can see the Associazione Nazionale Allevatori that are grouped by FEDANA, and as you can see, there is also Assonapa. Which is the role of Assonapa? Assonapa manage the Italian sheep and goat population through the herd books, and the herd books allow to do breed certification through the pedigree, conservation of the breed at risk through the inbreeding estimation and matings plan, improving milk and wool production through the estimation of breeding value, estimation of the inbreeding, and development of breeding plans. Furthermore, Assonapa promote the research with the aim to transfer a new selection and conservation tool into the field. And the general aim is to improve the production from quantity and quality point of view, to improve the morphology, and limit the increase of the inbreeding within the population, the breeds. All of these activities are based on the routine data collection that is carried on by Associazione Italiana Allevatori and Associazione Regionale Allevatori. In Italian, the routine data collection is called controllo funzionale. The controllo funzionale consists in an entrance of a technician with regular interval in a farm, and these technicians do data collection at animal level. The technician collect different data related to the reproductive events, individual production, and data related to the morphology of the animals. Here you can see the in the scheme on the left, you can see the data flow. All start from data collection. Data are transferred into the data bank of the breeding system, where there is the data bank of Assonapa. The data are managed and elaborated by our general office, and then services are emanated to the breeders through the data bank. On the right, sorry, you can see the main services of Assonapa. Among them, we have the pedigree management, we have the parentage analysis through DNA data, morphological evaluation, breed certification through the Airbook certificate, elaboration of genetic indexes, mating groups, and performance tests for meat sheep. We also do technical services for artificial insemination, and we organize show and breed contests. Okay, let's move to the Airbook. As I said before, with the new law, Decreto Legislativo number 52, we have a new definition of Airbook. The Airbook represents the tool for selection and conservation of the Italian sheep and goat breeds, and its aim is that to direct all activities for conservation and selection from a technical point of view. And the Airbook is the only tools for breed certification. As I said before, we have two type of genetic program: conservation, where the aim is to maintain the genetic variability within the population, and this is done through the inbreeding management, and in particular with the management of the matings. And the second type is selection, where the aim is to select animal in order to improve the performance of the animal, and selection is based on genetic or genomic evaluation that is the tool to that have the aim to that allow us to individuate the best animal, identify the best animal for a specific trait. The trait subjected to selection are planned into the Airbook. The Airbook works following a specific regulation. Assonapa has two regulations, one for goats and one for sheep. And the Airbooks works thanks to the Central Technical Commission, that is a commission that has the aim to evaluate all activities from a technical point of view. Works thanks to the General Office that organize all activity of selection and conservation. Thanks to breed experts that do the morphological evaluation. And the Airbooks works thanks to data collection. Okay, let's move to the breeds subjected to conservation program. One of the activity of Assonapa is monitoring of the Italian small ruminant population. As all activities, these activities starts from the data collection, start from controllo funzionale. The data collected at farm level are elaborated from Assonapa, and in particular, the number of male and female are calculated. Then the data are sent to the national focal point managed by CREA. The national focal point upgrade the DADIS data bank, and then for each breed, the risk status are defined. Taking into consideration the risk status for each breed, Assonapa apply two types of conservation strategies: in situ and ex situ. In situ conservation has the aim to manage the inbreeding at farm level through matings control, and ex situ conservation consists of collect and storage the semen in a semen bank. Here you can see an example of Bionda dell'Adamello goat, that is an autochthonous goat reared in this area near Adamello mountain. Here in the table you can see the number of animal that is around 1,600 animals. This goat is a dairy goat, and the main product are fatuli, the kid, and berna, that is a dried meat. I think that the characteristic of the breed we will see it this afternoon, and so I skip some slide. Okay, now let's see the breeds subjected to selection program. Here you can see the main goat and sheep subjected to selection program. Starting from the left, you can see the dairy sheep, that is Comisana, Massese, delle Langhe, Sarda. In the middle, you can see the main dairy goats, Camosciata delle Alpi and Sanen. And on the right, you can see the meat sheep, Merinizzata Italiana and Appenninica. Before to see an example of breed subjected to selection program, we have to understand how selection works. As all other activities, selection starts from data collection. So it is important the phenotype measurement, for example, the measurement of the production of milk or the quality of milk, fat and protein, morphology, or new phenotype of interest. Then it is important to know the parentage between animals. It is important parentage registration. We need to know the sire and dam of each animal. This because usually we are interested in female traits, and the female traits can be measured on female and not on male. So we can estimate the breeding value of a ram or a buck through the measurement of the phenotype on their daughters, granddaughters, or sister. And we connect phenotype to the respective male through the parentage, through the relationship existing between the animal. If we have a wrong registration of the sire dam, we have a wrong estimation of the breeding value. Then the estimation of the breeding value in Italian is called indice genetico. This is done subtracting from the phenotype all environmental effects that can be buildings, management, health, or nutrition. And then taking into consideration of the breeding value, we can identify the best animal for a specific trait that are made together in order to have a progeny with a higher breeding value. In recent years, in the last years, we had also a new step, the DNA collection. And thanks to the DNA analysis, we can do parentage check. This is important because one problem of sheep and goat sector is that farmers don't do mating group. So the sire and the dam could be unknown or could be wrong. So parentage check is very important to have an accurate breeding value. Thanks to the DNA analysis, we can select also for scrappy and Visna-Medi resistance, and we can also apply genomic selection. That means that we add to phenotype and parentage data, we add genomic data. Here there is an example to understand better what is a breeding value. Here you can see a distribution of a breeding value, a distribution with a mean equal to 100 and standard deviation equal to 10. And a breeding value is a value that tells us how much an animal produces from a genetic point of view, more or less than the population mean. We are interested to the best animal for a specific traits, for example, milk production. So we are interested to the animal located on the right of the mean population, highlighted in green. Selecting this animal and mate it together, we can have a superior offspring. Here you can see an example of two breeds subjected to selection program. The aim, in this case, the example is about Camosciata delle Alpi and Saanen. The aim of selection is morphology, in order to have a better udder and a better functional traits, and to improve milk production from quantity and quality point of view. In the table, you can see the tools that are available to select these two breeds. First of all, data collection, controllo funzionale. Morphological evaluation is very important to exclude from the selection animal with defects, morphological defects. Breed certification is very important. And these two tools are also applied to breed subjected to conservation program. Then we have parentage diagnosis, artificial insemination, genetic evaluation, the estimation of breeding value, and the development of mating program that allow to accoppiare, to mate animal together with the aim to maximize the breeding value and to while limit the increase of the inbreeding level. Here you can see the morphological traits that are collected in these two breeds. On the left, you can see the traits, the linear traits that are collected. We have udder profile, udder depth, teeth placement, rear udder attachment, and the hook distance. This trait have a linear score from 1 to 9, and in green is highlighted the best score for the trait. And the aim is to have all animal in this range. The score highlighted in red is considered defects. On the right in the table, you can see the defects that are collected, and the main are, for example, the unbalanced udder, accessory teeth, double teeth, umbilical hernia, hermaphroditism, or bite defects. Here you can see the list of estimated breeding value that are calculated in these two breeds. We have, for example, milk production, milk quality, protein and fat, in terms of kilograms and percentage. We have the 5 morphological traits that are recorded: hock distance, udder profile, teeth placement, rear udder attachment. We also have a combined combined index, for example, quality index that is composed by protein and fat kilograms. And we also have a global index that is composed by protein and fat, and the 5 index related to the morphology. So taking into consideration global index, you can select animal both for quality and for morphology. And furthermore, the kilograms of protein and fat are positive correlated to the milk production. So select for protein and fat kilograms, you select also for milk production. Thanks to our 2 projects, Cheese and Sheep and Goat, that are funded by the PSRN program, we now are developing new genomic breeding value. As I said before, On the left, you can see the traditional method to calculate breeding value, start from phenotypes and pedigree. Now we add a new step, and so we take into consideration also genomic data. What is the advantage with genomic selection is that we have a breeding value with more we calculate breeding value with more accuracy. We reduce the generational interval, and we improve the genetic gain. Here you can see an example of a genetic breeding value published. In this case, we have the example is on somatic cells. For each, we developed this index in Camosciata delle Alpi and Sanen, and we published it with a top 50 classification with the best 50 male or female for this trait. As you can see from the table, we included the classification, the animal ID, the breed, sex, year, the value of the breeding value, and its accuracy. For each new index that we developed, we also publish a little description about how the breeding values was calculated and how to interpret this index. What is the future? The future is to apply into the field genomic tools. In particular, for biodiversity conservation, the main tools is the estimation of genomic inbreeding, and in particular the estimation through the runoff homozygosity analysis. And for selection, the future is to apply genomic selection using DNA data, and on the right, in the scheme on the right, you can see the new traits that we now are analyzing. For example, we have the age at first parity, killing interval, somatic cell, nematode resistance, heat tolerance, lactation persistency, or productive life. Coming to the conclusion, why it is important to conserve the autochthonous breeds? Because selection operated from humans lead to the substitution of the traditional breed with the more specialized breed. And these lead to biodiversity loss. In the two boxes below, in the box on the left, I reported some strong point of these breeds. These breeds are very important because they represent an important source of genetic variability, and this is very important for food safety and food security. They are suitable for extensive farming that are characterized by low input, and they are very important because they are reared in a remote area, and they play an important role in landscape management. On the right, you can see some challenges that these breeds are facing, and we have a competition with specialized breed, climate change, and inbreeding. And thank you for your.
And if you want, we have other two two quiz.
One quiz, you can choose between sheep or goat. Goats.
Always goats.
Okay, we like goats.
This was the previous one. Okay.
The number is the same.
No, I don't think so. I will show you. Sì, sì. Yeah. Okay. Yeah, don't worry. You can already see the number here. Questo lo faccio vincere. No, okay. Menti punto. Menti. The number is 37515834.
Join. Sono...
Okay. Okay. Ok, ci sei dentro però? Sì. Ok, we'll start. I think. Let's start. No. Okay. Get ready.
Aspetta, dov'è?
Perché c'è Okay. It was a Saanen, the cosmopolitan goat from Switzerland. But alpine in the origin. Okay, very good. Okay. You should notice. Okay, very good. Okay. Well, Matteo is winning this time. Okay, this is a bit more difficult. They are almost from mountainous regions. Okay, the Nicastris is quite similar, but has longest hair and different horns and comes from Calabria. So the Nera di Verzasca is completely black. Just good to give some differences. Alessio, it was fast.
Adesso ho capito come fare.
Okay. Yeah. The most beautiful horns in the Italian panorama. Very good. It is a Giorgentana from Sicily. The snow was a trick. And the Sempione is white with lateral horns, whereas the only goat in Italy, at least, with those twisted horns is the Giorgentana. Okay. Get ready.
Ho sbagliato.
Ma come l'hai sbagliato?
No, giusto.
Okay, as I was saying, the Nera di Verzasca is completely black with short hair.
Si vede che
Okay. Let's see, because I don't know the order. Okay. Look also at the back. The color at the back, it is the same one. Yeah, the horns of the Val d'Ostana As we were saying, it can be of different colors, usually black or the chamoise, the camosciata that was at the back. But it is quite recognizable by the horns for the battle. Matteo is always faster than Alessio. Okay. We will forgive you. The Camosciata delle Alpi, the other cosmopolitan one, that is the classic chamois, I don't know the pronunciation, color, chamois. Okay. Almost done, I think.
Ah, questa non l'ho mai vista però.
No, l'hai sbagliata.
L'ho sbagliata.
It was the Sempione.
Allora,
Yeah, long fur, white fur, and quite
Oh.
big.
questo mi fa perdere la competizione.
Yes, I chose this because it was in the region.
Basta, non più.
Matteo, Banana Matteo is winning. Sail has gained a position. Okay, almost done. Last two. Yeah,
Questa
it can be black or it can be red.
ma ero lì pronto.
Yes, that is the Valesana.
Valesana.
It can be redneck or blackneck.
Tutto di nuovo, schiacciata. Vedi?
Yeah, only Matteo and Alessio. Well, it was not the easiest one, but it is quite recognizable. So for the future, you will know. Yes, the last one. You will know.
No, concentrate.
Okay. Oh, it's quite
Questa-
small. I'm sorry. But look at the horns.
Mi sa che mi ha battuto.
Okay. The Arabica has huge horns, but different from the Valdostana. And the winner is. No, sei stato veloce.
Ma ho sbagliato.
Matteo wins the competition.
Many compliments.
But compliments also to Sil because was not in the group and that is quite an
Okay,
advantage.
we want to conclude with some suggestion of book if you are interested in. So to win the next quiz, we can suggest the Atlas Atlante of Italian breeds. Maybe they can find the book.
Can you share the screen, please? Thank you.
And there are two versions, an old one and a new one. One that is dedicated to mammals, I think, and another one more recent that is dedicated to avian species. So this is the publication that you can consult to win the next quiz. And if you pass me the book, that book, that book, please. There is a very beautiful book of Francis that is domesticated and is the history of the domestication of the different species. A lot of different species, not only ruminant, but is very well written. And it reports all the study until the age of the publication of this book some years ago. And also he explained that domestication was winning lottery for the animals because they arrived until they continue to survive with us. Otherwise, a lot of wild animals are extinct because their habitat was destroyed by the human. And also there is a part that speak about the fact that also human is domesticated and so need a particular relationship between humans. So I suggest this. There is also the version, the audible version in English. Domesticated, Richard Francis. If you, it is the new one and is an audible also file. And it's really, so this is a lecture for pleasure if you are interested in this story, obviously. Okay, so thank you for your attention and thank you for the organizer of this.
Thank you very much, Alessia, too. for
Alessia, thank you, the staff.
managing all the meeting. And well, we hope that you enjoyed it. I think the speakers were very good, really. And well, now in the afternoon, we are going to see some goats, some local goat breeds. And well, bye-bye to everybody.